So you can hear me and see my title screen?
Yes.
That's great. Thank you. So thank you, Tom, for organizing this really interesting workshop. It's been great to hear all the talks about the stress response in different species. And I'm here to represent the rodent world. So I'll be talking about our work in rats in understanding the limbic neural substrates that regulate responses to social stressors in rats. And I'll start out sort of a bit broad with our research goals. The much of the work we do in the lab is based on this understanding that stressors, life stressors in humans that range from social stressors, stressors related to relationships, to bereavement, loss of employment, or range of stressors, that those can exacerbate or precipitate episodes of psychiatric disorders such as depression and anxiety, as well as substance abuse, and can also exacerbate or precipitate episodes of more systemic diseases like inflammatory bowel disease or obesity. So the way we try to address this goal is to look at the neural substrates, and this little picture is meant to represent a sagittal section of a rodent brain, is to look at the neural circuitry that controls the behavioral and physiological changes that occur as a consequence of stress. So while we know that stress exposure can have certain effects and impact individuals' functioning, we also know that there's a lot of variations between individuals, that there's individual differences. And another way to think about individual differences is that some individuals are more resilient to the effects of stress, while others are more vulnerable. There's many sources of these individual differences, including events that take place earlier in life that impact adulthood. So effects, experiences occurring in the prenatal life, neonatal, adolescent periods, coping strategies, which you heard about earlier today as well, genetics, and there's other sources of these individual differences. And we also look at the neural circuitry that is changed by these events, these sources of individual differences, and look at the behavioral and physiological consequences. Our third goal is to take some of the findings that we obtain in animals and to translate them into human subjects. And we do this through collaborations with other individuals here at the University of Pennsylvania and elsewhere. So what I'll talk about today is how coping strategies produce individual differences in response to social stress. So the model that we use for social stress is based on the resident intruder paradigm that was initially developed by Klaus Mischak in the late 70s. And we sort of modified it for repeated exposure. And in this case, what we have is, I don't know if you can see my cursor, but the albino experimental rat here, the Sprague Dawley rat, put into the cage of a larger, more aggressive resident rat, the Long Evans rat. So that's the home cage of the resident Long Evans rat. When you put the intruder in, initially these animals explore each other and sniff each other because they've never encountered each other before. So it's very natural for this kind of interaction to happen. But ultimately, with time, the resident rat, because he's dominant and it's his territory, it's his home cage, he defeats the intruder rat, as evidenced by the display of the supine defeat posture by the Sprague Dawley rat. When this happens, we separate the animals with a wire mesh barrier because we're not interested in injury and wounding that may occur with further interaction between the two animals. So they are separated for the remainder of the 30 minutes. So this entire interaction is just 30 minutes long and occurs every day for 5 to 7 days. And on each day, there is a different pairing. So the experimental rat is exposed to a novel resident rat each day. So there's no sort of habituation to consider here. So what we noticed as we were doing these studies is that the time it takes from being placed into the cage of the resident rat to the intruders showing defeat varied a lot across individuals. So looking at, 100 or more rats, if we just calculate the average latency averaged over those 5 to 7 days of defeat, we had a bimodal distribution. And so this is number of rats on the y-axis and the average latency. There were some animals that showed a relatively short latency to be defeated, and others had a relatively long latency to be defeated, with the middle being about 300 seconds, so about 5 minutes on the average. And if you plot out the daily defeat latencies of animals that we then subsequently identified as long or short latency, what you see in the second graph here is days on the bottom and the defeat latency. And you see in the first couple of days, everyone's getting defeated relatively rapidly. But over time, the short latencies tend to stay short. They're kind of pretty similar. It's the long latency animals that are increasing their latencies over time. So they're the ones that are changing more. When we think these differences between short and long latencies, we conceptualize them as coping strategies. The short latency animals are more submissive, and so they give up easily and show the defeat posture. The long latencies, if we quantify their upright postures and boxing postures, they show more of that. They're upright, kind of trying to fight back a little bit and resisting being defeated. So we think the difference between the short and long latencies is the manifestation of their coping strategies. If we look at behaviors after this week of defeat in the Poor Salt Poor Swim test, which I think many of you are probably familiar with, but it's often thought to be a test for sort of pro-depressive kind of behavior. If we look at the amount of time the animals spend in immobility, so they're placed in a tank of water and then exhibit either immobility or various types of activity, the short latency animals show higher immobility time. So they're more passive, again, consistent with their passive coping, more immobile, which is consistent with a pro-depressive kind of phenotype. If we look at them in the social interaction test, how much time do they prefer to spend near an animal versus an empty box? The short latency animals spend less time near a peer animal. And typically, two animals, as I said, who haven't found each other before are going to spend time interacting with one another. So the short latency animals show reduced social interaction time, indicating increased anxiety type behaviors. So we've also looked at other behaviors of these animals, including fear extinction and sleep disruptions. And all of this leads to our working hypothesis that the long latency animals are resilient to the effects of stress. They're typically not different than the control animals, whereas the short latency, more passive animals are vulnerable to the effects of stress. They're different than controls typically. So with that as a working hypothesis, what I'll tell you about today are two sets of studies that are focused on limbic structures. And again, this is a sagittal section of a rodent brain. And the two structures are the medial prefrontal cortex, shown on the top here, and the ventral hippocampus, which you heard about earlier, as well as important in anxiety behaviors. And the take-home message really is that both resilience and vulnerable substrates that we've been examining are based in neuroinflammatory processes. So the ventral hippocampus vulnerability is associated with increases in neuroinflammatory processes. In contrast, in the medial prefrontal cortex, resilience is associated with a reduction in neuroinflammatory processes. So these are the data that I'll tell you about in sort of summary form today. We're going to start with the ventral hippocampus. And as you heard earlier, it's intimately associated with anxiety-related behavior, whereas the dorsal hippocampus is more involved in spatial learning and spatial memory. And there are other regions that, come to mind when thinking about anxiety, such as the amygdala, for example. So in our previous studies, where we've looked at microRNAs and histone acetylation marks in the short versus long latency animals and controls. In the amygdala, we typically don't see a difference between short and long latency. There's a stress effect, to be sure. There's a defeat versus control effect, but no differences between the short and long latency animals. In contrast, in the ventral hippocampus, we've consistently seen differences between the animals we think of as vulnerable versus resilient. So we pursued the ventral hippocampus a little further here in studies that were published now a few years ago. So what we started with was the work of Jaya Pearson-Leary, who was a postdoc fellow in the lab, was using a discovery approach of looking at microRNAs, using microarrays of the ventral hippocampus. And the details here actually don't matter. We found 2 microRNAs that were different between short and long latency animals. They're listed here if you're interested. We then looked at what are the gene targets of these microRNAs and what are the functional pathways that might be regulated by these microRNAs. And these really led us to networks related to immune function, angiogenesis, and vascular remodeling. So genes associated with these networks are regulated by these 2 microRNAs. So Jaya looked at these aspects of immune function, angiogenesis, vascular remodeling in the ventral hippocampus further. And really here, what we're talking about is referred to as the neurovascular unit, shown up on the left here, of a blood vessel where blood vessels interact with neurons and astroglia and microglia. And so what we did was to look at measures amongst all these cells. Actually, we didn't really focus too much on the astroglia. Really, we're looking at neurons, microglia, and blood vessels. And this slide is sort of summarizing a lot of the work that Jaya did, which showed that vulnerable animals have increases in sort of chronic neuronal activity that's associated with vascular remodeling. So blood vessels become more complex, and maybe that's because they're supporting this elevation in basal neuronal activity. These vulnerable animals also show indices of inflammation, so there's more microglial activity, and there seems to be a disruption in blood-brain barrier permeability. It becomes more permeable. So all of these things are happening in the vulnerable animal, and that in these animals are also expressing the anxiety and depressive-like phenotypes. And so this is sort of a coordinated change happening in multiple cell types. And we then went on to show that if you inject a pro-inflammatory cytokine into the ventral hippocampus, you can mimic these aspects of vulnerability. If you inject an anti-inflammatory drug, you can prevent vulnerability from happening. So I won't show you those data, but this model is sort of, there's evidence behind these events being important for vulnerability. So in thinking about, how these changes are happening, they're not happening in isolation. They're occurring in the context of a cohesive integrated stress response, right? And we also know that central inflammatory changes are influenced by peripheral inflammatory changes. So one source of inflammatory immune molecules in the periphery is the gut microbiome. And John Cryan has really been a champion of gut microbiome in sort of neuroscience and stress. And this is just one way to think about it, is that in a healthy individual where the CNS function is normal, the individual's not stressed, they have a normal gut physiology. But in contrast here, in a stressed individual or somebody in a disease state, there are changes in behavior and cognition, et cetera, that are linked to abnormal gut function, particularly microbiota. So we then moved on to ask whether there are changes in gut microbiota occurring in our defeated animals. And so we simply started with characterizing the gut microbiome, and we collected a fecal sample 24 hours before a week of defeat, and then 24 hours after the last defeat, after we knew who was vulnerable and who was resilient based on their coping strategies. And so what I'm showing you here are population level graphs of the diversity of the microbiota in the 3 groups. So the controls are novel cage controls. They're placed in a novel cage every day. for 30 minutes while the other animals are being defeated. And the day 0 microbiome diversity is represented by the blue circles and the day 8 by the red circles. And what you see in the controls is just being put into a novel cage every day for 7 days does not change their gut microbiome. But you can see that in animals that are defeated and become short latency or long latency, their microbiome has shifted very distinctly in the short latency animals, and in most of the long latency animals, it's different than where it was day 0 pre-defeat. There's also differences in alpha diversity within an individual over time. And then we also looked at class levels and genus and species, and I won't get into those, but it looked like Clostridia, which are pro-inflammatory bacteria, seem to be higher in abundance in the vulnerable animals. So knowing that there are these distinct changes in gut microbiome, we asked what the contribution of the gut microbiome was to the resilient and vulnerable phenotype. So Jaya asked to try to answer this question using a fecal microbiome transplant approach. And so what we first did was to defeat animals, identify vulnerable, resilient, collect fecal samples from them, make a slurry and gavage those, so transplant those into naive animals. So 4 groups of naive animals that were not stressed at all in this experiment. So we had the first group were animals that were gavaged daily for 5 days with just PBS vehicle, a group that was gavaged with fecal samples that were collected from non-stress control, those novel cage control animals. And then the other 2 groups received microbiome transplants from vulnerable animals or from resilient animals. Okay, so 4 groups of animals, and then taken through the behavioral tests I've already told you about. And we also looked at what was happening in the ventral hippocampus. This graph is just to convince you that the microbiome transplants were successful. So if you look at the PBS control animals, day 0 is blue, day 6 is red. So this is how the microbiome changed from over the 5 days of receiving those oral gavages. And PBS vehicle alone does not change the microbiome, either does transplants from a non-novel cage control animal. But in naive animals that were receiving transplants from a short latency and a long latency, their microbiota shifted completely, suggesting that this oral gavage approach was successful in changing their microbiota. And we did this without any antibiotics, which we know can influence HPA activity and other aspects of the stress response by themselves. So that was a good thing. We were able to avoid giving these animals antibiotics. So what happened with these transplants? So if we look at behavior, this is immobility in the forced swim test. And you can see the animals that received transplants from short latency animals showed higher immobility in the forced swim test and a shorter latency to become immobile. So this is recapitulating the pro-depressive kind of phenotype that short latency animals I've shown you exhibit typically. Interestingly, there were no differences amongst the 4 groups in the social interaction test. When we looked in the brains, in the ventral hippocampus, the same animals that received the short latency fecal transplants showed more immobility, also showed more microglial activation. And in the blood, there was higher expression of IL-1 beta, the pro-inflammatory cytokine, as well as higher expression of S100 beta, which is a marker of blood-brain barrier permeability. So overall, so then there were no differences in body weight, there's no differences in neural activity or blood vessel densities. And we wouldn't necessarily expect neural changes because these animals were not stressed. So if we go back to the original model that I showed you about, which we used to describe how vulnerability occurs in the ventral hippocampus, now we can talk about what is the contribution of the gut microbiome to these substrates? So what does the stress gut contribute to vulnerability? Well, it does not contribute neural activity. There's no stress, there was no neural activity happening, and as a result, probably there's no vascular remodeling happening as a result of just a gut from a stressed animal. So the gut microbiome is not really influencing the neural activity and vascular remodeling in vulnerable animals, and as a result, it doesn't affect the anxiety behavior either. So what seems to be happening is that the complex neural and behavioral phenotype of vulnerable animals seem to be dissociable. The gut microbiome in a stressed animal is important for the microglial and immune activation that occurs in vulnerable animals and for their pro-depressive phenotype. But neural activity and vascular remodeling seem to be important for the anxiety phenotype. So we've now dissociated the anxiety from the depressive kinds of behaviors that happen in a vulnerable animal and shown a role for the gut microbiome in a subset of the vulnerable phenotype. So that is sort of our current understanding of how vulnerable animals become vulnerable, this role of the gut contributing to the ventral hippocampus, pro-inflammatory processes being activated to produce vulnerability. So at the same time, while we were working on the ventral hippocampus, we're also working on the medial prefrontal cortex to understand what might be happening there that might be different between the vulnerable and resilient animals. So in this case, we used a discovery approach, which was just using a targeted PCR array of a couple of hundred G protein coupled receptors to identify those that might be different between the short and long latency animals. And what we found was, of the 6 mRNAs that differed between short and long latency animals, only one had we never heard of before. So we pursued that because that was more interesting to us. So this was the sphingosine 1-phosphate 3 receptor, S1PR3, and it was increased in expression in the medial prefrontal cortex of the resilient animals in both stimulatory and inhibitory neurons, so generally increased in expression. It's one of 5 G protein coupled receptors for the endogenous ligand sphingosine 1-phosphate, which is a sphingolipid. And what S1PR3 is thought to do is to reduce inflammation in peripheral tissues, but we really don't know much about it in the brain even still. And based on our finding that S1PR3 expression was higher in resilient animals, we then tested whether if we reduce S1PR3, we can reduce resilience, and if we overexpress it, we can promote resilience in all stressed rats. So this is work by Brian Corbett, who was a postdoc fellow in the lab, and he used virally mediated knockdown of S1PR3 in the prefrontal cortex and looked at behavioral endpoints or virally mediated overexpression of S1PR3. And I won't show you those data. There's a fair amount, and it's published. But essentially what Brian showed was, indeed, if we knock down S1PR3, it reduces active coping strategies. It pushes all defeated animals towards reduction in active coping, increases in anxiety and depressive behaviors. In contrast, if you overexpress S1PR3, it pushes all animals towards a resilient phenotype. Okay, so S1PR3 is really promoting resilience, S1PR3 specifically in the medial prefrontal cortex. So of course, the question is, what is the mechanism of action of S1PR3? And we move forward here by looking at what S1PR3 is known to do in the periphery. And as I said, it decreases inflammation, particularly acting through IL-1 beta and TNF-alpha, which are pro-inflammatory cytokines. So Brian looked at the expression of these 2 cytokines in non-defeated and defeated rats with knockdown of S1PR3 or animals with scramble control. And he did this in 2 subdivisions of the medial prefrontal cortex, the prelimbic on the left and the infolimbic on the right. And what you see with the IL-1 beta is in the prelimbic, there's really no effective stress and no effective S1PR3 knockdown. When you get to the infralimbic, all animals with knockdown of S1PR3 have more expression of IL-1 beta, but stress has no impact on this effect of S1PR3. In contrast, TNF-alpha was more interesting because there was a stress effect there. And what we found is if you look at both the prelimbic and infralimbic, there were similar results. If you look at the two white bars, that shows you the effects of defeat. So defeat by itself increases TNF-alpha in the medial prefrontal cortex. And if you knock down S1PR3, TNF-alpha goes up even more. So there's a greater increase in TNF-alpha in a stressed animal if you knock down S1PR3. So we looked at the role of TNF-alpha in mediating S1PR3 effects in the medial prefrontal cortex, and how important is this TNF-alpha to the functions of S1PR3. So here in a series of studies, Brian used both viral and pharmacological means to reduce TNF-alpha, and I'm showing you the results of a pharmacological experiment where he injected either saline or infliximab to reduce TNF-alpha levels in the medial prefrontal cortex. So prior to daily defeat for 7 days, this TNF-alpha inhibitor or saline was injected into the medial prefrontal cortex. Animals went through defeat and went through the behaviors that I've shown you before. So just to go through the groups to make it clear, hopefully, there are 4 groups. If you look at the white bars, you see these are just normal animals, normal S1PR3, normal TNF-alpha, and the red bars are S1PR3 knockdown. Okay, so because S1PR3 is a resilience promoting molecule, if you knock it down, you reduce latency, you're pushing animals to the passive short latency phenotype, you're reducing their social interaction, you're increasing their immobility. So this is just replicating some work that I've referred to earlier. So S1PR3 knockdown pushes everyone against resilience towards vulnerability. If you just inject infliximab and manipulate TNF-alpha without stress, there's really no effect of that by alone. But if you take those S1PR3 knockdowns, so compare the second bar to the fourth bar, where you now are also knocking down TNF-alpha, you can see that knocking down TNF-alpha prevents the effects of S1PR3. So if you also inhibit TNF-alpha, you prevent the passive coping and the reduction in social interaction and the increases in immobility that happen if you just knock down S1PR3. So it looks like that S1PR3, so to summarize, S1PR3 is higher in the prefrontal cortex of resilient animals. And the way S1PR3 acts to promote resilience is by tamping down TNF-alpha in the medial prefrontal cortex. So this is sort of the model of the findings that I've talked about so far. In resilient individuals, they have higher S1PR3 compared to vulnerables. And that means that when these defeated animals don't show an increase in TNF-alpha, which is observed in the vulnerable animals, and that we think is important for the passive coping and social interaction. And this lack of TNF-alpha increase in the resilient animals is important for being resilient to the effects of stress. So there's sort of a couple of questions that emerge from this is, how is expression of S1PR3 regulated? Why is it going up in the resilient animals? How is it going up? And is this elevation S1PR3 pre-existing in the resilient animals? So the first question first, I don't know how much time I have left. I can cut through a slide if I'm.
I was going to say only a minute or two.
Okay. So the answer is glucocorticoid receptors, which are higher in density in the resilient individuals, and those in part are the reason why S1PR3s go up in resilient individuals. So is the S1PR3 that's higher in resilient animals, is that pre-existing? Well, in the brain, we can't address that question, but we can in blood. So we looked at blood levels of S1PR3, and we found that pre-existing differences were not there. So S1PR3 went up in the resilient animals in blood after the stress had happened. So there's no differences before stress. It's a result of stress, to the extent that blood represents what's going on in the prefrontal cortex. So we thought this was interesting because potentially S1PR3 could be a biomarker for resilience. And so we collaborated with our colleagues at the Philadelphia VA Medical Center to look at the blood of veterans who were combat exposed and did not develop PTSD versus veterans who did develop PTSD. And at 3 different time points, with the time points average shown here, in each case, individuals that did not develop PTSD had higher S1PR3. So essentially, we're recapitulating what happens in the animals. The higher the S1PR3, the lower the symptoms of PTSD shown here. Also lower re-experiencing and avoidance symptoms and arousal and nightmare frequency, depression. So the S1PR3 findings suggest that it might be a good marker for resilience to trauma or the effects of stress in humans as well. So one point I want to highlight here is that vulnerability is not less resilience, and resilience isn't less vulnerability. These are unique processes. Looks like they're regulated by unique substrates. And finally, just to end with this slide, because many of you have heard of multiple substrates of resilience, how is that possible that there can be so many substrates of resilience or vulnerability? And this is to kind of suggest that it depends on the stressor, it depends on the individual. So on the left here, when an individual is exposed more to cognitive types of stressors, the limbic substrates of resilience come into play more and impact the outcomes. In response to more metabolic and physiological stressors, you may have more hypothalamic and brainstem substrates that predominate. And also across individuals, our past experiences, epigenetics, and genetics can influence the dominance of these pathways and will determine what substrates dominate in one individual over another. So there's a lot of things to work out in understanding resilience still. And I wanted to thank some of the lab members that contributed and our funding agencies. Thank you.
All right, thank you very much, Seema. I think we should open up for some questions if anybody has any questions. I'll start with one. You kind of answered it with the S1PR3 in the end, but I was wondering from the beginning, this difference between long latency and short latency rats, is this a pre-existing, even if the S1PR3 isn't pre-existing, that is responding to the stress, there must be some difference between these rats ahead of time. Have you ever tested them? So if you, for example, if you do the behavioral testing, the forced swim test and all that, if you did that before the exposure to the repeated defeat stress, would that predict whether they end up being LL or SL rats? Or does it require that week of stress in order to get those phenotypes?
Right. So we have done some of those behaviors before we stress them, and we haven't seen any differences. We've done some fear conditioning. There's post-defeat fear conditioning changes that happen, but different things happen pre-defeat. So there's not an easy way to predict behaviorally. We did identify and publish that there were some microRNAs in circulation that was different pre-stress. So there's potentially this a couple of microRNAs in blood that could help you predict who's going to go on to be vulnerable or resilient. So there is that potential there. Of course, there's genetic factors, and we haven't done any kind of sequencing to see if there pre-existing genetic differences, I would expect these are outbred rats, so I expect that there's going to be some genetics that explains this, but I think it really takes the stress to make them fully express those phenotypes.
Thank you. Spock. Sorry. Is he not able to speak? Hold on. Trying to find you.
He's muted.
Sorry, this is me. Now you can speak.
I'm back again. My apologies. I just switched to my phone. Seema, lovely presentation. I was curious. I know it's difficult to disentangle this, but I'm curious about the temperament of these animals, because I know in the dog world, folks have spoken about a different temperament playing a kind of an interlocking role with the resilience of the animal. Is there ways in which you could test that, or is there any other data, like if you put them in novel environments, or what's their response to, I don't know, auditory or cues or things like that?
Seema, you're muted.
Temperament. So I don't really know how to address that, other than if you look at some of the passive and active coping literature, there are various other sequelae of those coping strategies that we haven't fully examined, like autonomic nervous, like a predominance of sympathetic over parasympathetic kind of thing. Those I think are probably there, but we haven't fully looked at other kinds of behaviors that might get at the temperament. There are some fear conditioning differences I was mentioning earlier that also pre-exist. There may be some inherent differences in freezing fear responses. But yeah, we haven't fully examined those. It's very interesting.
Great, thank you. All right, in the to stick to time, or mostly to time, I would then like to now invite the final speaker of this afternoon, Carrie Figdor. Carrie is going to be the one talk that is not about neuroanatomy, or at least not